Cardiotoxicity is heart damage that arises from certain cancer treatments or drugs. It can develop years after cancer treatment, especially in adults who received cancer treatment during childhood. Certain types of cancer treatments have a higher risk for cardiotoxicity. Cardiotoxicity may make it harder for your heart to pump blood throughout your body as it should. In severe cases, it may lead to cardiomyopathy, a heart muscle condition that makes it harder for your heart to pump blood. Cardiotoxicity can affect anyone who has had cancer treatment. It is more likely in those who have taken specific drugs or had radiation therapy to the chest. It's also somewhat common in adults who had cancer treatment as a child. It's difficult to determine the exact rate of cardiotoxicity in adults who had cancer treatment as an adult. But some groups estimate up to 20% of this population may develop heart problems, with up to 7% to 10% having cardiomyopathy or heart failure. Blocking a protein known as CDK7 could prevent heart damage associated with a commonly used cancer chemotherapy medication, according to a study led by scientists at Washington State University. Importantly, the researchers also found that inhibiting CDK7 could help enhance the medication's cancer kill. Based on an animal model, the study findings could provide a foundation for future treatment strategies to reduce chemotherapy-related heart toxicity and increase treatment effectiveness. This could ultimately help increase the lifespan of people with cancer. Heart damage related to chemotherapy treatment can surface decades after treatment and can result in heart attacks, heart failure, cardiomyopathy, and other types of heart disease. Published in the journal Cardiovascular Research, the WC study focused on doxorubicin, a chemotherapy drug used to treat breast cancer, lymphoma, leukemia, and other cancers. Capable of killing a wide range of cancer cells, doxorubicin and other similar chemotherapy medications are known to be toxic to the heart. Despite this toxicity, the drug still sees a lot of use. Doxorubicin remains the mainstay treatment for certain cancer types, for which targeted therapies or other better treatments are not available. Scientists have been working to unravel the underlying mechanisms of doxorubicin-induced heart toxicity to make the use of doxorubicin safer for patients who rely on the drug. This new study builds on findings from earlier research that showed that doxorubicin activates a protein known as CDK2. That protein then activates another known as FOXO1, which causes heart cells to die. Researchers take a closer look at CDK7, a protein that helps fuel cell growth and has been shown to play a role in the development of cancer. The researchers found that CDK7 activated CDK2, which set off the chain of molecular signals that eventually led to heart cell death. They also showed that mice that lacked the CDK7 gene were protected from doxorubicin-induced heart toxicity. Next, they used a CDK7 inhibitor drug known as THC1 to block the protein's activity and examine the impact on heart health and cancer growth. A similar inhibitor is currently being tested as an anti-cancer drug in clinical trials, but its effect on the heart is still not clear. This is the first time to study the effect of THC1 on the heart and on tumor growth in the same model, and what we found is that this CDK7 inhibitor drug can increase heart function and at the same time inhibit tumor growth. Though more research is needed, the researchers said their findings suggest that combining doxorubicin and THC1 could help prevent heart damage and increase the effectiveness of chemotherapy treatment. The researchers' next step is to test the effect of THC1 on heart damage and cancer growth in younger mice and follow them longer. This would more closely mimic long-term doxorubicin-induced heart toxicity seen in childhood cancer survivors. They also plan to look at other proteins that may somehow be involved in the signaling pathway that underlies doxorubicin-related heart damage. Thank you for watching. If you've enjoyed our videos, subscribe to our channel for there's more to come.